So we're finished with the osteoblasts, which were the simple cuboidal layer of densely stained cells present in the periosteum and the endosteum, and they were secreting bone matrix, right? Now, after their synthetic activity is completed, osteoblasts actually differentiate. So you know the process of differentiation in which certain genes are turned off, certain genes are turned on, and they differentiate into newer cells, right? So osteoblasts also, after their synthetic activity, their bone matrix production is complete, they differentiate into some inactive cells called osteocytes. And we know what osteocytes are. They are the cells of the bone matrix, right? The cells present inside the bone matrix, inside their own lacunae, and they were connected with each other by little small canals called canaliculi, right? So that's the osteocytes. Osteocytes are basically the most abundant cells in bone tissue, and they exist within their own lacunae. Obviously, they are surrounded by bone matrix. We have looked at the diagram before, and they are interconnected by canaliculi. This interconnection of canaliculi results in exchange of materials, exchange of information through gap junctions. Very, very important point. So if somebody questioned you about a cell type in bone tissue that has gap junctions, then you should be able to say that those cells are actually osteocytes, right? Osteocytes have long dendritic processes that extend into the canaliculi and have gap junctions which allow them to exchange information with other cells of the with neighboring cells of that particular bone tissue. The reason why bone, the hard rigid bone, the bone matrix is referred to as an alive tissue is because actually of osteocytes. So bone matrix does contain the osteocytes, and they are alive cells, and they are supplied by blood vessels that we have already looked at, right? And the Haversian canals and the perforating canals and so on. And they have this important function for bone maintenance and remodeling. So basically they're able to detect healthy and unhealthy stresses. They're able to detect increase in stress, and they're able to detect decrease in stress. And depending on the stress received, they release some paracrine and endocrine signaling for bone development. So they may release some factors that would stimulate bone development and increase bone density. Or due to decrease of stress, they might release some factors, or they might stop releasing factors that result in decrease in bone density, more resorption of bone rather than production, right? And that is the function of osteocytes. A good example of this is in the astronauts. In astronauts, which are people in space, obviously, vaguely putting it, because they function under a very low or almost close to no gravity, they have this lack of bone stress, right? Because they're not bearing any weight, right? So this lack of bone stress actually leads to a decreased stimulation or decreased stimulus of the osteocytes, and this results in more bone resorption and decreased bone density. So as osteocytes are cells of the bone tissue and they keep the bone alive, then death of an osteocyte would actually lead to resorption of bone around that particular lacuna, all right? Okay, so we have finished the osteoblasts, the osteocytes, right? And those were all cells of the periosteum and the endosteum. Next up, we have the osteoclasts. Osteoclasts are very important, high-yield key players of bone tissue, of bone chapter, of bone histology, okay? Osteoclasts are actually the cells that are responsible for bone resorption. We've been discussing bone resorption all this time. And osteoclasts are actually the cells that perform it. So osteoclasts are, let's have a look at this diagram right here. Osteoclasts are giant cells. These are very large. They can easily be identified by their huge size. And second of all, they also have multiple nuclei, right? So first of all, they're giant. That's histological feature number one, is that they are giant cells, right? So if you see periosteum or endosteum histological slide and microscopic slide, and you see that there's a giant cell with multiple nucleus or nuclei, then that would be an osteoclast. And the function that it performs is bone resorption. And also, in some slides you might be able to see that they have this cavity right here in contact with the bone matrix where it basically performs the resorption of bone, and this cavity is called a resorption cavity, or you could also call it a house-shaped lacunae or resorption cavities. As I mentioned in the very first video of this series, which is basically before you start histology with breast medicine, I mentioned that all of these diagrams are taken from Junqueira's basic histology book. And we have permission to use these diagrams, and I encourage everyone to go to the Amazon affiliate link that I have in the description below to purchase this book, because it's a very good read and it will really help you develop concepts of histology. All right, so now let's look at where the osteoclasts originate from. Where do they come from? Well, unlike the osteocytes and the osteoblasts, which came from the mesenchymal stem cells, the osteoclasts actually come from the hematopoietic stem cell, right? HSCs, hematopoietic stem cells in the bone marrow. We've already studied them. Hematopoietic stem cells, they have a chapter of their own, hematopoiesis or hemopoiesis, coming up next. In which we study the details of them, but basically in our bone marrow we have hematopoietic stem cells. These are multipotent stem cells that are able to divide into certain different types of cells. And one of those cells that they divide into are monocytes, right? Monocytes are precursors of macrophages, right? And then there's MCSF released, macrophage colony stimulating factor, which is released to stimulate or differentiate monocytes into macrophages, different kinds of macrophages, depending on the location where they receive this stimulating factor. So one place in which that happens is actually the bone tissue. Monocytes that travel through blood into the bone tissue receive this macrophage colony stimulating factor to stimulate the development or differentiation of monocytes present there, and they develop into macrophages or osteoclasts of the bone. Osteoclasts of the bone. And these osteoclasts then go ahead and start doing their function. So these are large multinucleated cells that have resorption cavities. And I have this diagram right here which shows that here's an osteoclast which has multiple nuclei, and it's a giant cell, and it is associated with a blood capillary near it, and we'll look at how that plays a role in just a bit. And then it has this seal zone at the side and a cavity right here in the very middle, and this is actually the resorption cavity, the house-shaped lacunae, singular lacuna, where all the resorption actually occurs and goes on. So let's zoom this in and try to work out what's going on. This resorption cavity has actually a ruffled border on the osteoblast side, so it has this ruffled sort of, kind of like villi, you'd say. Right? It has numerous folds, and these numerous folds actually increase the surface area, and it plays a role in the absorption of stuff that is released from matrix. We'll look at that in just a bit. But what happens is that inside this ruffled border, there's actually some pumps that are pumping protons into this resorption cavity. So this resorption cavity, this house-shaped lacuna, becomes very, very acidic because protons are being pumped into the resorption cavity. Similarly, another thing that is being pumped is MMPs, matrix MMPs, matrix metalloproteinases, right? MMPs right here. Low pH and concentrated MMPs. MMPs are matrix metalloproteinases, which basically are matrix degrading enzymes. So low pH and matrix metalloproteinases together play a role in digesting, breaking down the bone matrix and resulting in bone resorption. So when the matrix is digested, and we studied that the bone matrix is mainly made up of calcium and phosphate ions, then calcium and phosphate ions from the hydroxyapatite, calcium and phosphate ions are released, and this ruffled border, which increases the surface area, actually helps absorb these calcium ions back into the osteoclast. So calcium and phosphate ions then are absorbed into the osteoclast, and from the osteoclast they go to the nearest blood vessel, which is right here, right? And then the level of calcium and phosphate ions in the blood go up. This function of resorption of osteoclasts is actually stimulated by parathyroid hormone, which is released by the parathyroid gland, which is a gland present behind the thyroid gland, right? And it is actually downregulated by calcitonin, and we'll also look at this in the endocrine chapter as well. So in conclusion, we can say that osteoclasts are actually cells that come from the hematopoietic stem cell, and they are large, giant, multinucleated cells that have these resorption cavities, these house-shaped lacunae, which are able to basically absorb bone matrix. And the way it happens is because they have some proton pumps and they have some matrix metalloproteinases that are being sent into the resorption cavities. This low pH due to the protons and the concentrated MMPs result in degradation of the bone matrix in which it is contact with, and it results in the digestion of bone matrix results in release of calcium and phosphate ions that then are absorbed by the ruffle border, and it goes into the nearest blood capillary to increase calcium and phosphate levels in the blood. And all of this function is actually upregulated, stimulated by parathyroid hormone. We have a parathyroid gland behind the thyroid gland, and what it does is that it is able to detect low calcium levels, low phosphate levels, and it secretes the parathyroid hormone, and it goes through the blood to our bone tissues and stimulates this entire process. High calcium levels would actually stimulate the parathyroid gland to release calcitonin and decrease the production of parathyroid hormone, PTH. So calcitonin would downregulate the activity of the osteoclast, and that's pretty much it with the osteoclast physiology. So now that we understand what the function of osteoclast is and how it functions with regards to its different features like the sealing zone and the ruffle border of the house-shaped lacunae and its ability to pump protons and matrix metalloproteinases to digest the matrix proteins, we can now try to understand some clinical correlates, okay? And number one, we have actually osteopetrosis. Osteopetrosis is when there's actually a defective ruffle border, right? A defective ruffle border. We said that defective ruffle border plays a role in bone resorption. So a defective ruffle border would not only result in less secretion of protons and matrix proteinases into the resorption cavity, but also less resorption of the released calcium and phosphate ions. So it basically, overall, we could say that it leads to less bone resorption. And so there's a higher bone density, right? And higher bone density would mean that there's more bone matrix and less space for the bone marrow, right? Less medullary cavity, more bone matrix. So it results in lesser hemopoiesis or lesser hematopoiesis, and basically less production of blood cells. So it results in anemia and low blood cell, white blood cell count. Next up, we have osteoporosis, which is due to estrogen deficiency. Due to estrogen deficiency, it is better explained in physiology, but due to estrogen deficiency, we have more bone resorption, and it results in low bone mineral density and results in basically very weak and brittle bones. It's a very tragic disease in which there's very weak and brittle bones, and those bones are actually very easily fractured. Osteoporosis is a disease which is almost 4 times more common in women than in men.